In this video, we'll review the transformation process used to insert plasmids into the E. coli bacteria you'll be using in this project. As you know, plasmids are circular pieces of DNA used to drive the production of specific proteins. These plasmids include promoters, operators, and associated elements like genes that encode for specific antibiotic resistance proteins and the target protein. In our project, the antibiotic resistance gene will be for ampicillin, and the target protein will be insulin.
This protein lets the bacteria live in the presence of a particular antibiotic, in this case, ampicillin. During the transformation process, we selected only the E. coli that had taken up the plasmid, made the antibiotic resistance protein, and will be able to survive in the media after the ampicillin is added.
The plasmid also includes a gene that ultimately will result in synthesis of our target protein, insulin. This gene is driven by the T7 promoter we also inserted into the plasmid.
To help make sense of this, we'll take an even closer look at the relevant parts of one of the plasmids inside our E. coli. These bacteria synthesize many copies of a special enzyme called the T7 RNA polymerase that selectively bind to the T7 promoter in the plasmid. This results in the synthesis of the target protein. The gene responsible for the T7 RNA polymerase is controlled by a region in the bacteria's DNA called the lac promoter. The lac promoter is controlled by an operator region just downstream that normally is silenced by the repressor protein. This prevents production of the protein encoded on the plasmid. Good so far?
We're with you, Diener. At the right time, we'll add a chemical called IPTG to the media. IPTG, which is structurally similar to lactose, removes the repressor protein, which then allows the T7 RNA polymerase to do its job. As a result, the target gene DNA is transcribed into mRNA. This ultimately results in production of insulin. And as you like to say, Dean, voila!